welcome also on my behalf. I'll start by sharing my screen. Okay, so now I picture that you are seeing my slides. All right, so this lecture is intended as a primer to EEG and MEG signals, and also to get an idea of why we might want to use EEG and MEG to study Alzheimer's disease. Okay. So I will talk first about the basis of the MEG-EEG signals, how they arise, the neurophysiology underlying these signals, and also a bit about how we can measure these signals. What kind of devices do we need to measure it? I will focus on oscillatory activity in the brain, spontaneous activity in the brain, and how they might be forming networks in the brain. And finally, just very briefly, I will mention some effects that take place in oscillatory activity when we age normally, and also in what has been observed in neurodegeneration. Okay, so information flows in the brain as electrical signals. And electrical signals in any wire generates an electromagnetic field. And this happens also in the brain, even if the electromagnetic fields that are produced by neural activity are very weak. And this is what we can measure with MEG and EEG. With MEG, we can measure the magnetic field caused by neural activity outside the head. And with EEG, we can measure the potential at the surface of the scalp related to these same neural currents. So the benefit of MEG and EEG is that we get a direct measure of neural activity through this electromagnetic field, and also that we can measure it with excellent time resolution, so we can look at a very sharp, fast events that happen to some outside event or stimulation, or we can look at ongoing activity at different frequencies, at the direct neural frequencies that they take place. So first, a bit about how these or what type of currents we have in the brain. So when looking at MEG and EEG, it is beneficial to separate the neural currents into, or the currents that flow in the brain, into primary currents and volume currents. The primary current is the current that we are interested in. It flows inside the cell and in its immediate vicinity. Whereas volume currents are passive ohmic currents that flow in the extracellular space. And they arise because there can be no buildup of a charge in a conducting medium. So therefore, we have these return currents flowing. So let's have a look at the currents at a synapse. So in the axon, we have action potentials that are propagating along the axon. And at these action potential, so that we have currents flowing into the cell, and there is a wave of current propagating in front of the action potential, and a second wave behind the action potential. So forming a quadrupolar field. And at the synapse, we have at the postsynaptic part of the synapse, we have a dipolar field where currents are flowing in just one direction. And the currents or the electromagnetic field are then caused by all currents that are flowing. So when it comes to the magnetic field, we can use a rule of thumb to get an idea of what type of a magnetic field is caused by the currents. So if we use a right-hand rule, and we know that there is a current flowing in the direction of our thumb, then we know that the magnetic field produced will be perpendicular to this current and flow in the direction of our fingers around the current. The volume currents will then fill up the conducting space and flow backwards from the currents to even out the charges. So these two constructs are useful, the primary and the volume currents. Another useful construct when we talk about currents in the brain is a current dipole. So any primary current can be described by a point-like current source. These dipoles have a strength and an orientation, but they have no extent, they are point-like. And the dipole moment then is described by the strength and orientation. And now the primary currents and any currents sum up linearly, meaning that any primary current distribution in the brain can be represented as a combination of current dipoles. And this will be useful then when trying to deduce from where in the brain the activation comes from. Okay, so how do MEG and EEG then relate to each other? So we know from electromagnetic field theory that magnetic and electric fields are oriented perpendicular to each other. So now if we simulate a dipolar source in the brain and have a look at the field patterns that are generated outside the brain, we can see for the MEG field patterns, a field according to our rule of thumb, right-hand rule. So the currents flowing in between the two extrema and on the side of each of the source, we have magnetic fields either flowing out or flowing into the brain. Now the same dipolar source in EEG is perpendicular to the MEG field patterns. We have a positive deflection in front of, above the dipole, and a negative deflection behind the dipole. And you can also see that the field patterns are smoother for the EEG, so the spatial resolution is lower. Okay, these are typical topographic maps. Okay, but in order to understand our signals, it's important to know what currents cause our signals. So it's usually said that EEG and MEG signals are generated by synchronous postsynaptic activity in the apical dendrites of pyramidal cells. And I will open up this statement a bit. So first of all, what are the apical dendrites of the pyramidal cells? Well, pyramidal cells are large excitatory neurons with somas concentrated to layers 2 and 3 and 5 in the cortex. They have a long apical dendrites that are oriented in parallel to each other, meaning that then currents flowing in these apical dendrites will be summing up. Okay, so those were the pyramidal cells. So we're saying that it is the postsynaptic potentials that generate the MEG and EEG signals, but why not the action potentials? They are, after all, larger in amplitude. Well, it is here the timing of these currents is essential. So in order to see the signal outside the head, we need a lot of signal summation, and the action potentials are fast. They last about 1 to 2 milliseconds. So the probability of these currents summing up at the same time is much lower. So there will be less temporal summation of the signal. Whereas the postsynaptic potentials, even if they are of smaller amplitude, they are slower, so their temporal summation is more likely. And secondly, I talked about quadrupolar and dipolar fields earlier and the action potential propagating along the axon. Now, it can be shown that a quadrupole causes an electromagnetic field that attenuates very fast with distance. So meaning that when we look at the data from the sensors outside the head, the signal will be very weak. Whereas the dipolar field also attenuates with distance, but not as fast as the action potential. So it's more likely for us to see these types of currents outside the head. Okay, so summing, how many neurons would we then need for us to see a signal outside the head? So I talked already about currents summing up linearly. And the go-to paper to find out how many neurons we would need in order to see a signal is a paper by Murakami and Okada, where they made simulations for both MEG and EEG signals. And I show here an example for MEG signals. So in their simulations, they found that the dipole moment for typical pyramidal cells is about 0.2 to 0.9 picoampere meters. And we know from our measurements that a typical MEG response would be about 10 nanoampere meters. So they estimated that about 50,000 synchronously active neurons would be sufficient to generate an MEG signal. And here it was very relevant for their simulations that these apical dendrites of the pyramidal neurons are indeed oriented in parallel so that they sum up. Now, of course, this is an estimate of how many neurons we would need to see a signal, but it doesn't say anything about cancellation. So it's much more difficult to estimate what type of cancellation is there in the cortex. But all in all, we can say that MEG and EEG measures the synchronous activity of tens of thousands of synchronously active pyramidal neurons. So what about whether the currents that we measure are excitatory or inhibitory? Can we see this or tell based on MEG and EEG signals? It turns out no. Only based on our MEG-EEG signals, we can't really say that the excitation would be excitatory or inhibitory. So here we have an illustration of excitatory and inhibitory synapses and what happens to the current. So we have the pyramidal neuron, and in this first example, there is an excitatory synapse in the apical dendrite where many of the excitatory synapses are located. And we see a typical current flowing towards the soma. Now, for an inhibitory synapse, quite often located at the soma. So now the inhibitory synapse will cause neural currents of opposite polarity at the soma, and the net result will be the same within the cell, currents flowing towards the soma. And again, if we have an excitatory synapse at the soma, the currents will be flowing in the opposite direction. So it's not so simple to tell what type of synaptic signal we are measuring with MEG and EEG. Okay, so one other very important aspect when it comes to whether we can pick up a signal is how the cortex is oriented with respect to the surface of the skull. So we know that the pyramidal cell dendrites are oriented perpendicular to the cortical surface. But of course, the cortical surface is organized into gyri and sulci, and these, the cortex in itself can be oriented either tangentially or radially with respect to the surface of the actual head. And this affects especially MEG signals. So let's take a special case that we have a participant whose head is a perfect sphere. In this case, we know from electromagnetic theory that a radial source will not cause a magnetic field outside this perfect sphere at all. And the reason is symmetry. So the volume currents that flow in the media around this radial source will perfectly cancel out the original primary current. So there will be no net magnetic field. In a tangential source, a source that would be located inside a sulcus. So here the symmetry is broken, and the volume currents that flow have a different path that they can take on each side of this primary current. And this type of a source configuration will cause a magnetic field that is measurable outside the head. Now, EEG measures also the radial component because there we are measuring directly the volume current at a specific location at the surface of the head. Okay, so this was the case for a participant with a spherical head, but real participants do not have perfect sphere heads. But it is still true that the detection probability for a radial-like source is much lower. But we see here in these simulations in red those brain regions that cause very low detection probability, or where we need a much stronger signal to detect the source. And what we see is that there are parts of the cortex that we cannot see with MEG, but what is really visible from this image is that deep sources generate very weak signals. Okay, so what about EEG? So I already said earlier that EEG has a lower spatial resolution than MEG. And why is that? Well, in EEG, the problem is that the signal is distorted, smoothed, low-pass filtered by the poorly conducting skull. So we know that the conductivity of soft tissue, brain, cerebellum, scalp, is much higher than that of a skull when we make models of our of the conductivity in the head, we typically model the conductivity in the skull to be 50 times smaller than for soft tissue. And this then causes the lower spatial resolution of EEG compared to MEG, because the magnetic field passes through the skull. So MEG gives complementary information then over EEG. All right, so that was a few words about how the signal is generated. But how do we then measure it? So modern MEG systems use SQUID sensors. These are superconducting quantum interference devices that can measure very weak magnetic fields. However, in order to be superconducting and have these properties to measure weak magnetic fields, they need to be immersed in liquid helium. So we need a slightly larger and bulkier system to accommodate then this liquid helium. And also these systems are very, very sensitive to noise, so they are usually situated within a shielded room. Okay, so the SQUID sensors we have systems that have different types of sensors. We call them magnetometers or gradiometers. Here, the SQUID in itself is the same, but what is different between our different sensors are the pickup coils. So we need a pickup coil that brings the magnetic field to our SQUID sensor. And these can be constructed in different ways. So a magnetometer measures the magnetic field directly, and it consists of a single loop that then measures the magnetic field that flows through it, and it shows the maximum signal where the magnetic field is strongest, so a little bit on the side from the actual source. To cancel out noise, we can also use different geometries for the pickup coils or the flux transformers. We can use gradiometers that measure the difference in magnetic field, and they consist of two loops that are wound in opposite directions. An axial gradiometer has a single loop close to the brain and a second loop in the opposite direction, slightly further away. And now the idea is that environmental noise will be flowing through both loops, whereas the brain signal will be flowing or picked up by the loop that is closer to the head. And this way, the background noise will be cancelled out. And in our systems that we use within this project, we have magnetometers and then planar gradiometers that then have two loops next to each other. And these planar gradiometers measure the derivative of the magnetic field and show a maximum signal right above this current source. So what about EEG? With EEG, we use electrodes that are attached to the surface of the scalp, and we try to get good conductance between the scalp and our electrodes. I show here a couple of EEG sensor systems, a cap with, I think, 64 electrodes, and then a higher density cap with 128 electrodes. There are different types of EEG systems. And one thing to keep in mind when you look at EEG data is the naming convention for these electrodes. So in the 1020 naming convention system, we have odd numbers for the electrodes on the left, even numbers on the right, and then the electrodes are named based on where they are located. F for frontal, and P parietal, and O for occipital, and so on. And here I have a very, very simplified circuitry for EEG, considering an amplifier. And what is important to remember in EEG recordings is that it measures the difference in signal between two electrodes, an electrode and a reference electrode, or if we have a bipolar montage, so the difference between neighboring electrodes. And looking at our data, it matters what the reference was. Just look at topographic maps, so we can re-reference our data, use the reference that was used during the recording, or calculate what the data would look like if the reference would be somewhere else. And this affects how our data looks like. A typical way to re-reference the data would be to use the average signal as a reference. So what about MEG and EEG? How do these signals relate to each other? Here we have data, visual data. So there are flickers shown either to the right or the left, visual hemifield, many repetitions, and this is an averaged evoked response, how the brain has reacted to visual input. So, and now we can see immediately from these topographic plots where the signal is coming from. So remember the right-hand rule for MEG. The current flows in the direction of thumb and the magnetic field around it. So we can immediately see from the magnetometer maps that the current would be situated between the two extrema here in the back of the head. The gradiometers measured the strongest signal right above the current, so we have one extrema showing exactly where the activation came from, and the EEG topography is perpendicular to the magnetometers, which you can see here as well, and slightly blurred. Okay, so let's have a look a bit at the signals. So both EEG and MEG provide multi-dimensional time series data. And from this, we can extract multiple measures of brain function. We can look at so-called evoked responses, just averaged to many repetitions of some external event. We can look at oscillatory activity, just the frequency components of the signals. We can look at coherence between the signals and some external reference signal, for example, muscle signals, or we can look at functional connectivity, synchronization, coherence between different brain regions. But what we most likely will get stuck with immediately when looking at the data are the artifacts. And it's always good to know your enemy. What do they look like? And here are some typical artifacts from MEG recordings. Here we see a clear blink, eye blinks cause artifacts in our data because the electrical signals from the eyes are much larger than anything that we will ever measure from the brain. We see so a blink, an eye movement, so that's a saccade. The subject is looking from the right to the left. We see muscular tension, so high frequency muscular artifacts, respiration, so slow movements. If the participant is actually moving, then there will be even clearer drifts like this. We see artifacts related to heartbeat, ECG, and these are particularly visible in MEG magnetometers. Or if we forgot to ask the participant to remove for example, watches or anything like that, then we might have additional artifacts. So knowing just how your artifacts look like is always useful. Okay, so I will talk a bit about ongoing oscillatory activity. These are spontaneous rhythms that are modulated by different tasks, and they were first measured with EEG by Hansberger in 1929. These are clear signals that show up as soon as you start measuring. And with MEG, the first signals were measured in 1972. And here you can see how these 10 Hertz alpha activations are modulated when you are opening or closing your eyes. So this oscillatory activity has been given names. So we have canonical frequency bands, delta, theta, alpha, beta, gamma, where alpha and beta are the ones that are most clearly visible in awake normal participants. And the spectrum of this oscillatory activity shows this periodic oscillatory activity, but also this typical aperiodic or 1/f components, and exhibits this type of power law signals. So what does then these oscillations do? Well, we know that these oscillations modulate during different tasks. The alpha oscillation is attenuated, for example, or it becomes stronger when you close your eyes. It becomes stronger when you're feeling drowsy. It is attenuated in visual tasks, also if you're just imagining that something visual is going on. And it's also modulated with attention. So for example, if you attend to a visual event in your left hemifield, then alpha in the ipsilateral hemisphere will be increased, and contralaterally it will be attenuated. And these types of modulations have then led to a suggestion that alpha is related to inhibition, because there is stronger alpha when you're not doing anything visual, and the alpha activation is decreased when you're actually performing a visual task. So what about the beta oscillations, the 20 Hertz oscillations? Well, they are mostly seen in the sensory motor cortex, and they are modulated by motor, tactile, proprioceptive tasks. And you can see a typical attenuation of beta activation after a movement, here in this picture, a passive movement, and then a rebound. And it's the idea is that it reflects the excitability of the sensory motor cortex. And there is also evidence for functional roles of these beta frequencies, so that, for example, if there are beta frequency bursts before a very, very weak touch, then this affects the probability whether you can actually detect this very faint touch or not. And there is strong correlation between the MEG and EEG signals in these types of tasks. Okay, so we have talked a bit about the oscillations, but how do they come about? Well, there are many different mechanisms that could cause these types of oscillations. So oscillatory activity is an inherent property of neural networks. So it has been shown that it is enough to have local networks in layer 5 of the cortex to cause alpha oscillations. So in these animal recordings, it has been shown that oscillations can be generated locally within neural circuits. A very common mechanism for creating gamma frequencies are a so-called ping network, so pyramidal interneuron gamma type of network. And here many theoretical accounts have shown that these interneurons, networks with interneurons, shape the gamma frequency oscillations. And they work so that if there is an excitatory activation affecting the inhibitory neurons, they will become active and in turn inhibit the excitatory neurons for a certain refractory period, after which the excitatory neuron can again fire, and so on and so on and so on. So these are also local circuits within the cortex that have the property to cause gamma frequency oscillations. Then there are models that also investigate the role of input from the thalamus on these oscillations, especially during sleep. It's known that thalamic input is important for alpha frequency oscillations. And here I have just an example where a thalamocortical model explained quite well observed pharmacological manipulations on alpha and gamma oscillations. So clearly these cortical subcortical circuits also play a role in the generation of cortical oscillations. So what about then networks? So those were local oscillations. Well, there are many theoretical accounts for how oscillations could be relevant also for network functioning. And one of these theories is the how coherence between or communication through coherence could affect information flow in the brain. So we know that oscillatory activity is an intrinsic property of neuronal groups. And if we have coherence among different neural groups that are oscillating, then because the neurons are more responsive at a given time point, then this creates efficient time windows when communication is better. So these types of mechanisms could allow for flexibility in for example, in routing information. An area could be differently connected during different tasks, even if the anatomical wiring is the same. Now, of course, if there are some pathological mechanisms causing abnormalities in the oscillations, then this also means that information flow across networks will be impaired. And this brings us then to how the oscillations might be changing either normally over age or in neurodegeneration. So here I have an illustration of how the alpha peak frequency changes as we grow older in normal age. So here the color shows not amplitudes but frequencies, so the peak frequency of the alpha frequency. And here are young adults, our typical university students that we usually have as subjects. Now, if we move forward in age, we can see that the peak frequency is getting slower. You see that the color is changing, meaning that the oscillation is slower. And this is a known phenomenon from EEG. So what about neurodegeneration? Well, we talked about excitatory and inhibitory synapses and how they maintain the balance in neural networks. Now we know in Alzheimer's disease that synapses are affected. So both amyloid beta and tau aggregations cause problems at the synapses. So for example, amyloid beta accumulation can cause neural hyperexcitability, and this can also be seen as more epileptiform activity in mouse models. And tau concentrations causing synapse loss also. Then one hypothesis for the cognitive deficits in Alzheimer's disease is that network dysfunction is causing it. So there have been many reports on how the EEG and MEG signals change in AD pathology. But what are the most typical findings? Well, first of all, the frequencies slow down, so there are more slower frequencies are more prominent in the spectrum. The complexity of the signal is reduced. We look at nonlinear effects, so we can see reduced complexity of the signal overall. Synchronization between brain regions is affected, and this has been reported in many studies in EEG and also in MEG and at multiple frequencies. Okay, so this brings us to the end of this primer. And we talked about how the signal was generated in postsynaptic potentials, through postsynaptic potentials, and also how they were measured, and a little bit about oscillatory activity and how that is affected in aging and in Alzheimer's disease. So if you have questions, then please go ahead.